something out here which holds the key to the secret of good whiskey. Yeast for a successful fermentation. We all know about the yeast that makes bread rise, that puts bubbles in the beer. But there are also wild yeasts all about us. In the grass, in the air, in the flowers. Thousands of different strains. To find yeasts with efficient fermentation characteristics and a unique flavor contribution, Seagram went back to the source. Researchers gathered thousands of samples of wild native yeasts, traveling the country from east to west. The wild yeasts found in the pollen of these flowers were a long way from a usual pure culture, but they would be taken back to the distillery, where the long process of selection, incubation, and purification would single out the best. From the thousands of specimens, 5, after years of testing, proved to be of a quality and character suitable for fermentation of Seagram whiskey. Samples of these precious yeasts are freeze-dried and kept in suspended animation in vacuum tubes, one of the most closely guarded secrets of an industry. When yeast is required for fermentation, one of these tiny vials is broken open and the dried cells brought back to life in a nutrient solution. Here they will thrive and grow under the right conditions. Yeast reproduces rapidly and will soon use up a flask of sugar medium. It grows by ingesting sugar, in this case the maltose or sucrose molecules in the converted grain mash. Here we see how the yeast cell takes in the maltose molecule through its permeable cell walls. Enzymes in the yeast break down the sugars, allowing the cell to grow and divide. When all the molecules have been used up, the flask is full of yeast cells. This in turn is added to a larger tank of grain mash called the donut tub. Here, the yeast will work for 12 or 14 hours until it has grown to sufficient volume to inoculate a whole fermenter. In the fermenting room, the vast tubs of cooked grain mash are waiting, and the yeast wastes no time, as the vigorous bubbling testifies. Under the surface, there is equally hectic activity. The grain mash contains millions of sugar molecules, all of which must eventually be consumed by the yeast cells. The distiller's interest in this is twofold. Firstly, flavoring elements, called congeners, will be created as the yeast enzymes break down the molecules, important to the flavor of the future whiskey. And then, of course, there is the production of alcohol, created by the fermentation process, along with carbon dioxide gas, all that busy bubbling in the fermenter. After 3 to 5 days, the bubbling will cease. This means that the yeast has done its job and is no longer active. Where there were maltose molecules before, now there is an 8% alcohol solution, known in the industry as distiller's beer. Its destination, the still house, where the fermented mash will be distilled into a pure, crystal clear spirit. The third stage in the making of a Canadian whiskey.